Okay, this is a lecture that corresponds to chapter 16 on biochemistry. Just to start with a basic definition of biochemistry. So biochemistry is the chemistry of all living things and life processes. So in this chapter, we're going to be talking about some of the key biochemical molecules that have to do with life and living processes. Just a little sort of basic introduction. If we're going to talk about the cell is the basic component of all living organisms. And we're not going to get super in detail, but in general, cells are enclosed by a cell membrane, and that membrane helps to regulate what can pass through the cell and out of the cell. In addition, you have other types of cells called plant cells, and those have a cell wall that's composed of something called cellulose, which we'll talk about in a little bit. Here we have an example of a plant cell in green and an animal cell in blue. And these cells in general have a lot of similar components. There's the nucleus where the genetic material is held, and that controls heredity. There's ribosomes. These are the structures where protein synthesis occurs. There's mitochondria. That's the cell structure where energy production occurs. And then in the plant cells, you have chloroplasts, and that's where photosynthesis occurs. And the reason why we're sort of doing a little introduction to the parts of the cell is that we are going to be talking a lot about the biochemical molecules that are either made by the cell or needed for the cell to grow. To start with, we're going to talk about where energy comes from in biological systems. So green plants contain those chloroplasts that I mentioned in the last slide. And those are capable of taking the energy from the sun and storing it as chemical energy in something called glucose molecules or sugar. Plant cells are also capable of converting carbohydrate molecules, which we'll talk about, into fat molecules. And some can even convert them into proteins. Animals on their own cannot produce their own energy. They have to obtain energy either by eating plants or other animals that eat plants. So you can see the basic process of photosynthesis takes carbon dioxide, CO2, and water. And in the process, in the plant, it absorbs sunlight. That's the energy that produces sugar, C6H12O6, that's glucose, and also oxygen. So that's the process of photosynthesis. Energy in animals comes from consuming food. And we're going to talk about the three basic biochemical molecules that make up nutrients, where energy can be obtained. And those are carbohydrates, fats, and proteins. Before I get to that, just a basic definition of the process of metabolism. Metabolism is a series of chemical reactions that help keep a cell alive. And there are two basic types of metabolic reactions. You can have a catabolic reaction, and that's where molecules are broken down in order to produce energy. And you can have an anabolic reaction, and that's the process of building or synthesizing molecules. And both of these things occur in the human body. We break down molecules, we break down glucose molecules in order to produce energy, but we also use energy to take smaller molecules and build bigger molecules. So just to start with a little bit about carbohydrates, there are different types of carbohydrates. There are simple carbohydrates and complex carbohydrates. The monosaccharides, glucose, fructose, and the disaccharides, sucrose, are the most common dietary sugars, and these are considered simple carbohydrates. The reason why they're called simple carbohydrates is that they are able to be absorbed directly into the bloodstream. They don't need to be broken down or hydrolyzed first. And that applies to glucose and fructose, but not sucrose. Sucrose actually does need to be broken down. So they're called simple carbohydrates because they can be accessed, turned into blood sugar right away once you consume them. Complex carbohydrates need to be broken down by the body. One example of a complex carbohydrate is starch. Starch is broken down into glucose, And another example is cellulose. And remember, I mentioned cellulose earlier as the component of the cell wall in plant cells. Humans lack the ability to break down cellulose. So in general, cellulose is not considered a carbohydrate for humans. It acts more as a fiber. As I mentioned on the last slide, complex carbohydrates are called complex because it takes the body longer to break them down. So the complex carbohydrate and a simple carbohydrate are classified based on how quickly it's digested and absorbed. Glycogen is the mean by which animals will store glucose. It's stored in the liver, and the body can store around 2,000 calories of glucose in the form of glycogen in the liver. When the body is depleted of glucose, the body can then go through a process called glycogenesis, where the glycogen that's being stored in the liver is converted to glucose in the bloodstream. Once the body runs out of glycogen, they will immediately be unable to perform, and that state is commonly described as hitting the wall. There are some techniques that are used in order to maximize glycogen stores. These include things like carb loading, so consuming large amounts of carbohydrates so the body is forced to store them as glycogen that can be used later. Also consuming glucose gels. So if you have depleted your body of glucose and glycogen, consuming a glucose gel will give your body access to that glucose in the bloodstream. Another form of taking advantage of the way the body can store and use glucose is the ketogenic diet. In the ketogenic diet, carbohydrates are often avoided so that when the body does need to begin to turn to other sources of energy, there's no glycogen there to be broken down, so the body has to move to breaking down fat as a form of energy. Fats are used for energy, insulation, and cell membrane. They're digested in the small intestine and generally stored in adipose tissue that are called fat deposits. Fat deposits are located around major organs and beneath the skin. Diets that are high in fat and cholesterol have been implicated in something called arteriosclerosis and cardiovascular disease. You can see in this slide some common sources of fats include things like bacon and cream and oils and butter. Cholesterol is a steroid-like compound that's common to animal tissue. So anytime you consume any fats that are derived from animal products, those contain cholesterol. Cholesterol are the compounds that clog arteries, forming plaques, leading to that arteriosclerosis that I mentioned before. Cholesterol is carried into the blood by something called lipoproteins, and these lipoproteins are categorized according to their density. The two that are the ones that we're going to talk about briefly are low density LDL lipoproteins and high density HDL lipoproteins. Low density lipoproteins carry cholesterol from the liver to the blood. So they take the cholesterol that's been stored in the liver and bring it to the blood. These are the proteins that are responsible for the formation of those plaques that clog the arteries. So LDLs are called bad cholesterol. HDL lipoproteins carry cholesterol from the blood to the liver, and so they're considered good cholesterol because they help to lower overall cholesterol levels. LDL levels can be lowered with medications, also losing weight, quitting smoking, becoming more active. But overall, you can be genetically predisposed to have either high levels of HDL or high levels of LDL. So there is some component of just a genetic, maybe you are more prone to have high LDL, which would mean high blood cholesterol levels. Diets high in saturated fats and trans fats have been linked to those high LDL levels in the blood. So overall, limiting the amount of saturated fat and also trans fat in one's diet is generally recommended. Saturated fats and unsaturated fats are not all that different structurally. So you can see to the right, there's a saturated fat and an unsaturated fat. Saturated fats are called saturated because they're saturated with hydrogens. So it's just a straight chain, essentially, of carbon and hydrogens. Unsaturated fats have double bonds randomly throughout the structure, which results in that kind of kinked structure. As you can see, the one that's bent is an unsaturated fat. Saturated fats, because they are essentially straight lines, that allows them to stack really nice together, really tightly together, which means they're generally solids at room temperature. Examples of saturated fats: meats, butter, and dairy products. So those sources of fats that come from animals, like I mentioned before, those tend to be high in cholesterol and increase the level of the LDL lipoproteins that take the cholesterol from the liver to the bloodstream. Unsaturated fats, these, because they're kinked, they can't really get close together, and that makes them liquid at room temperature. Examples of unsaturated fats are things like vegetable oils. These have been shown to increase the HDL lipoproteins, which is taking the cholesterol from the blood and bringing it down to the liver. Trans fats are formed when unsaturated fats are hydrogenated, which essentially means they're forced to break those double bonds, adding hydrogens to make the margarines more solid at room temperature. Another type of biomolecule that we're going to talk about are proteins. Proteins are made up of little things called amino acids. A bunch of amino acids together make a peptide, and a series of peptides together make a protein. There are 20 different types of amino acids, and these are important for the body to use because they use them to make proteins in order to be used for growth and repair. Of those 20 different amino acids, the body can synthesize all but 9 of them. The remaining 9 amino acids are called essential amino acids, and they're essential because they are needed from the diet. So they're essential to be received from the diet. If we're talking about consumption of protein, which we'll talk about in a later chapter, in general, it's thought that the human body needs about 0.8 grams of protein per kilogram of body weight. Protein deficiency can be a big problem. It can lead to slowed growth and development, discoloration of the skin, hair, and also a distended and swollen abdomen. Structure of a protein generally looks like this. The term primary structure is the name given for a chain of amino acids. Once that chain of amino acids becomes to take shape, it can take shape in 2 different ways, either as something called an alpha helix or a beta pleated sheet. That is called the secondary structure. When we go then to the tertiary structure, that is when there is some folding upon itself. And then the final structure is the quaternary structure, and that's a multitude of protein or polypeptide chains that have come together to make an overall protein. Enzymes are biological molecules. Most enzymes are proteins. These are highly specific molecules that help to catalyze a reaction or a group of reaction. Catalysts, remember, are things that speed up the rate of a reaction. They're not used up in the process, they just help facilitate the reaction in order for it to occur faster. Substrate is the term given for the reactant molecule. So the molecule that the enzyme is helping to facilitate the reaction with is the substrate. As I said before, these are specific. The substrate will bind to something called an active site on the enzyme. When that happens, you've formed something called an enzyme-substrate complex. That's the name given for the substrate and enzyme together. The enzyme will help facilitate the reaction within the substrate and then leaving the products at the end. After the reaction has been done, the enzyme returns to its original form, ready to help catalyze another reaction. The action of enzymes can be inhibited, which essentially means blocked. A molecule can bind to the enzyme or protein, the enzyme protein at another site other than the active site. It could also bind to the active site as well. That could cause the protein to change shape and prevent that substrate from fitting in. Diabetic test strips use two enzymes to measure blood sugar. One enzyme catalyzes the reaction with glucose, producing hydrogen peroxide as a byproduct. The other enzyme helps break down that hydrogen peroxide in order to produce a color change to indicate the level of blood sugar. Enzymes can also be monitored to diagnose liver damage and heart damage, and they can be used to break up clots after a heart attack, or even to increase clotting to treat hemophilia. The final category of biological molecules that I'm going to mention are nucleic acids. Nucleic acids serve as information and control centers of the cell. They come in two different forms: deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. Both DNA and RNA contain long chains that are called nucleotide. And you can see the picture in the upper right, that's a nucleotide. A nucleotide has a sugar molecule, a phosphate molecule, and then something called a heterocyclic amine base. So essentially you have a bunch of those molecules that are linked together, making DNA or RNA. As you may be aware, DNA is the hereditary molecule within animals. It helps to carry our genetic information. So that was a quick summary of the chemistry behind some important biological molecules, and that is the end of chapter 16.